So, hello everyone. This is the eighth problem solving session for the course Environmental Chemistry and Microbiology. The course instructors are Professor Anjali Pal and Professor Sudha Goyal from IIT Kharagpur, and I'm Shrija Ghosh, a PMRF from IIT Kanpur. So, in today's session, we'll be talking about several different topics related to microbiology. We started with introduction of microbiology in the previous session. So today we'll be discussing more about cell chemistry and the macromolecules that are present inside the cells. Uh, so we'll be discussing certain questions related to your eighth week's assignment, and feel free to ask any doubt if you have in these questions, or if you have any doubt in general in any of the topics in this week's session. I'm here to clear your doubts. So let's start the session, everyone. So I'll be asking certain questions which, if you know the answer, please write the answers in the chat box and then we can have a discussion. If you have any doubt in any of the questions, please ask. Then also we can have a discussion with everyone so that we'll be able to understand the topic better. Okay? So starting the session now, starting with the first question. Question number one. Bacterial cells can be stained with dash to reveal the presence of lipid inclusions. Option A, safranin. Option B, methylene blue. Option C, trypan blue. And option D, Sudan dyes. Bacterial cells can be stained with dash to reveal the presence of lipid inclusions. What stain do we use to stain the lipid inclusions? Option A, safranin. Option B, methylene blue. Option C, trypan blue. And option D, Sudan dyes. Please write the answers in the chat box. I request all of you to write the answers in the chat box so that we can have a discussion. I have one answer in the chat box, option B. What about others? Even if you don't know the answer, just try to answer something so that we can discuss later if you are right or wrong. Anybody else? Okay, so the correct answer for this question is option D, Sudan dyes. So, safranin, methylene blue, and trypan blue, these are also dyes that are used for staining bacterial cells, but each dye has its own purpose and each dye has its own characteristic features which give a specific color to a specific structure. Okay, so Sudan dyes have a high affinity to fats, and therefore these dyes are used to reveal the presence of lipid inclusions. So lipids are basically made of fatty acids, right? So we need dyes that can bind to these fats, or this that can bind to these lipids so that we can stain the lipid inclusions. So for that, these Sudan dyes, since they have a high affinity for fats, we use these dyes uh, to stain the lipid inclusions. Other than that, we have we have safranin, methylene blue, and trypan blue, right? So safranin is a dye that is used in gram staining. We'll discuss gram staining in detail later. So it gives a pink color to the cells. It has a reddish pink color and it gives pinkish color to the cells. It generally stains the cells completely. Uh, so it is generally used in gram staining. So when we heat fix the cell, it completely stains the cell pink. If uh, the cells are rod, you will see pink rod. If the cells are spherical, then you will see pink circles, like that under the microscope. Then comes methylene blue. Methylene blue is a positively charged dye. So it can bind to DNA. Okay, so methylene blue can bind to the DNA since DNA has a negative charge, right? So DNA has a negative charge. So methylene blue being a positively charged dye, it can bind to the DNA and stain the nucleic acid. Okay. Trypan blue, on the other hand, it is a negatively charged dye. I'll just write here. Methylene blue is a positively charged dye and trypan blue is a negatively charged dye. So trypan blue is generally used for checking the cell viability. How do we check cell viability? So if our, so let's say I have mixture of live cells and dead cells. Okay, the dead cells will lose its shape and dead cells will be the cell wall of the dead cell or the cell membrane of the dead cell won't be intact. Okay, so when we stain with trypan blue, since trypan blue is a negatively charged dye, the membrane is also negatively charged. So trypan blue cannot enter the live cells. Okay, it cannot enter the live cells, it can only enter the dead cells, and dead cells have certain pores present on the cell surface, so dead cells can take up these trypan blue. So when you will observe under the microscope, the uh, dead cells will appear blue in color. So dead cells will appear completely blue like this, whereas live cells will appear colorless. So live cells will appear colorless and against a faint blue background. Okay, so live cells will appear colorless and dead cells will appear like completely blue, filled with blue color. Okay, so this is how trypan blue staining looks like. Methylene blue also looks kind of similar to this since it stains the DNA inside the cell. Saffron will look pinkish in color, and Sudan dyes, they generally have a black or blue color, and they will stain the lipid inclusion specifically. It has a high affinity towards fat, so it will stain all the lipids. Okay. I hope that's clear to everybody. Anybody has any doubt, please ask. Okay, then we'll move on to the next question. Cell size and cell number can be measured using option A hemocytometer, option B Coulter counter, option C Petrov-Hauser counting chamber, or option D none of these. Cell size and cell number can be measured using hemocytometer, Coulter counter, Petrov-Hauser counting chamber, or none of these. Which of the following can be used to measure the cell size and cell number? Okay, Ipsmita is saying option C. What about others? Anybody else wants to answer? Which of the following can be used to measure both cell size and cell number? Okay. Dilip Kumar says option B, Coulter counter. Okay. Anyone else wants to answer? So option B is the correct answer. So the other two options, hemocytometer and Petroff-Hauser counting chamber, both of these are similar instrument and they are basically used to count the number of cells, but cell size cannot be determined by these uh, two equipments. Coulter counter can be used for determining the cell size and cell number. So what happens is this is how Coulter counter looks like. Like this is a diagrammatic representation of Coulter counter. So we have the sample where we have the cells. Okay. Now cells will be of all different sizes. Okay. Now what will happen? The from the sample, these cells will pass through this small hole and there will be an interference in the current. So now if the particle is big, the interference will be more. If the particle is small, the interference will be less. Okay. So how many times the interference is there? That will give the number of cells. And what is the intensity, pulse of the current, that will give us the size of the cells. Okay. So it can count the cells as well as it can measure the cell size. Okay. So it can measure both cell number and cell size. Now we have Petroff-Hauser counting chamber, or we had the hemocytometer. So this is how the slide looks like. Okay. So we have two different sides from which we can load the samples. We put a cover slip over this, and from this groove that you can see, I hope everyone can see. I'll just use a pen. Yeah. So this groove you can see, right? From this point, we use a micropipette and we slowly inject our sample so that the sample is completely filled in this. And this part is basically, when you look under a microscope, it looks like this. It has multiple grids on it. Okay. Now, so there are two types of grids you can see. The outer ones have bigger squares and the inner ones have smaller squares, right? So the outer ones are used for counting bigger cells like mammalian cell counting. We are using these outer corner squares and we use the inner corner square for counting smaller cells like a bacterial cell. Okay, so Petroff-Hauser chamber is generally referred, like this term is generally referred when we are doing the bacterial cell count and we are using these smaller squares. So the length of both the larger and the smaller square is different. So this is 1 mm by 1 mm. Okay, this is 1 mm and this is also 1 mm. Okay, whereas this small square that you see, I'll just highlight one square. So you see this square, right? This one small square, this corresponds to 0.2 mm into 0.2 mm. Basically, length and breadth is 0.2 mm. Okay. So, when we load the sample, If we are loading the sample here, the sample volume would be the sample volume would be 1 mm, 1 mm into 1 mm. This is the length and breadth, and the height is 0.1. Okay, 0.1 mm. When we put the cover slip on top of this, the height difference between the glass slide and the cover slip, this is 0.1 mm. So the volume comes around 0.1 mm cube. Okay. The volume comes around 0.1 mm cube. Whereas in this case, you will have the height comes for Petrov Hauser chamber, the height is 0.02 mm. Okay, 0.02 mm. So if you see this whole square, the fifth quadrant that is there, so fifth quadrant, the whole square would be 1 mm by 1 mm. Okay, each small square is 0.2 mm by 0.2 mm, and the whole fifth quadrant, the volume of fifth quadrant would be 1 mm into 1 mm into 0.02 mm. So this will be 0.02 mm cube. So both the equipments are same. It's just the difference between the volumes that are there. Okay, so since the bacterial cells are smaller in size, we cannot count. There will be many cells in these bigger quadrants. So we choose one small square so that we can count the number of cells in that one small square, and then we'll take four of these squares, and then we can take an average of the number of cells and divide it by the volume. So volume we are getting from here. So we'll get the number of cells per ml. Okay. So that is how we use a Petrov-Hauser chamber or a hemocytometer. Okay. This is clear. Anybody has any doubt in this? Okay. If anyone has any doubt, please ask uh, regarding this. If you have a doubt how to count the cells. So you can see here, we can count the cells in one of these uh, squares, and this is how the cells will be. The cells will be there outside the square also, but we will only count the cells that are inside the square. Okay. And generally the rule is we can count if let's say this is the square. If let's say this is the square, so there can be cells that are touching the edge of the square, right? There can be cells that are touching the edge of the square along with the cells that are present inside the square. So the ones that are present inside the square, you will definitely count them. But what about the cells that are touching the edge of the square? So the rule is, if it is touching the left edge or the top edge, we'll count it. If it is touching the right edge or the bottom edge, we won't count it. So these two won't be counted, but these two will be counted. Okay. So that is the rule of counting the cells in a hemocytometer or a Petrov-Hauser chamber. Okay, that's clear. Okay, let's move on to the next question then. Question number 3. Most microbial structures and enzymes are composed of option A lipids, option B proteins, option C carbohydrates, and option D carbohydrates and lipids. Most microbial structures and enzymes are composed of option A lipids, option B proteins, option C carbohydrates, and option D lipids and carbohydrate. Requesting all of you to write the answers in the chat box. have only one answer, option B. What about others? Anyone else wants to answer? Okay, I have one more answer, option B. So everyone is going with protein. So that is the correct answer, option B. So protein forms up to 55% of the dry weight of bacterial cells. So when I say dry weight, that means cells have a water content, but out of the dry weight, that is excluding the water content, whatever is there, from that uh, dry weight, 55% is proteins. The virus coat is also made of proteins that envelops the nucleic acid. So even the virus that are the smallest microbes, we can say, they are also made of proteins which envelops the nucleic acid, that is the genetic material. Okay, so proteins is the major macromolecule that is found. So this is the list of the macromolecules. So protein, we have different types of macromolecules. We have proteins, we have polysaccharides, lipids, liposaccharides, DNA, RNA, etc. Okay. Now proteins have the major share. These macromolecules comprises of 96% of the dry weight. Out of that 96, proteins have the major share. They are contributing to the 55% of the dry weight of a cell. Polysaccharides are contributing towards 5%. Lipids are contributing towards 9.1% of dry weight. Then lipopolysaccharides are contributing to 3.4% of the dry weight of a cell. Then DNA 3.1 and RNA 20.5. So after protein, the most major fraction of macromolecule is RNA. Okay, then we have some monomers present in the cell apart from these macromolecules, the monomers of these macromolecules. So amino acids and precursors, which are responsible for the protein formation, they are also there in 0.5%. Then sugars and precursors. So these are the precursors of polysaccharides and lipopolysaccharides. Then we have nucleotides and precursors, which are the precursors of DNA and RNA. So all of these are also present in smaller quantities. And then we have inorganic ions, that is 1% of the dry weight. Okay, the protein is present in the major amount, and then comes the RNA, and then comes other micromolecules. Okay, that's clear to everyone. Okay, let's move on to the next question then. Which of the following organelles contain DNA, divide, and possess some degree of autonomy? Which of the following organelles contain DNA, divide, and possess some degree of autonomy? Option A, Golgi apparatus. Option B, ribosome. Option C, mitochondria. Option D, peroxisome. Which of the following organelles contain DNA, divide, and possess some degree of autonomy? Option A, Golgi apparatus. Option B, ribosome. Option C, mitochondria. And option D, peroxisome. Okay, we have one answer, option C. What about others? Everybody try to answer. Anyone else wants to answer? Which of the following organelles contain DNA, divide and possess some degree of autonomy? Golgi apparatus, ribosome, mitochondria, peroxisome. Okay, we have two different answers, option C and option D. Mitochondria and peroxisomes. Okay, what about others? Okay, so option C is the correct answer, mitochondria. So cellular organelles like mitochondria and chloroplast. So chloroplast in case of plant cells and mitochondria in case of animal cells have evolved from specialized bacteria which survived endocytosis by other cells and got incorporated into the cytoplasm of the cell. And hence they are known as semi-autonomous organelles. So during evolution, during the course of evolution, there uh, had been certain bacteria that were engulfed by larger cells, and then those bacteria evolved into an organelle that survived in the cell and became mitochondria or chloroplast. Okay, so these have DNA and these have the ability to divide. Okay, that's clear. So mitochondria and chloroplast, both of them have DNA and they have the ability to divide also. So they are kind of semi-autonomous organelles. Okay, that's clear to everyone. We are moving on to the next question. Question number 5. Calculate the wavelength of light if diameter of the smallest resolvable object size is 250 nanometer and the specimen is observed with 100x oil immersion lens having numerical aperture of 1.2. Calculate the wavelength if the diameter of the smallest resolvable object size is 250 nanometer and the specimen is observed with 100x oil immersion lens having numerical aperture of 1.2. You have to calculate the wavelength, the resolution is given, and the numerical aperture is given. Okay. Anybody knows the formula? What formula should we use? If you know the formula, apply the formula and calculate, or at least write the formula in the chat box. Uh, can everyone not see my slides, or is it your network problem? Can you check? Can someone confirm if my slides are visible? Okay, I would suggest you to disconnect the call and then connect back. Maybe you have some network issue, okay? If it is visible to others, then you might have some network issue. Anyone knows the answer or knows how to solve this? Otherwise, I'll tell you the formula and I would request you to solve it on your own first. So resolution is equal to the wavelength divided by the 2 times numerical aperture. So we have the resolution given as 250 nanometer. We have to calculate this wavelength, and we have the numerical aperture as 1.2. So use this formula and solve it and write the answer in the chat box. Oh. Dilip has solved it and he's saying 360 micrometer. Okay. Anyone else? Anyone else is able to solve it? Okay, option D. What about others? I've written the formula. You just have to use your calculators. It's not very difficult. At least try to solve it, everybody. Is anyone else able to solve this? So 600 is the correct answer. 600 is the correct answer. The units in the options are given wrong. I'll change this. This will be nanometer only. Both of them will be nanometer only. The diameter of the smallest object resolvable is 0.5 lambda, and then 0.5 lambda by numerical aperture. That is, I wrote the 2 in the denominator. If you take it in the numerator, it will be 0.5. Lambda is the wavelength. So you had the resolution as 250 nanometer, numerical aperture as 1.2. So wavelength would be 600. Okay. Is this clear to everyone? If anyone has any doubt, please ask. Okay, then moving on to the next question. Question number 6. Sorry. Transmission electron microscope is best to observe. Option A, a stained bacterial smear. Option B, unstained live tissue when it is desirable to see some intracellular structure. Option C, intercellular detail of a cell that is 1 micrometer long. And option D, a sample that emits light when illuminated with UV light. Transmission electron microscope is best to observe. Option A, stained bacterial smear. Option B, unstained live tissue when it is desirable to see some intracellular structure. Option C, intercellular details of the cell that is 1 micrometer long. And option D, sample that emits light when illuminated with UV light. Transmission electron microscope. It is best to observe what? Okay, we have two answers, option D and option C. What about others? Okay, we have two answers as option C, one as option D. Let's see the correct answer. The correct answer is option C, intercellular details of a cell. So transmission electron microscope, by the name itself, you can see it has electrons as the uh, light source. So basically electron beam is transmitted through the sample. So there are two types of electron microscopy commonly used for structure determination. One is transmission electron microscope, other one is scanning electron microscope. So T stands for transmission, and here the S stands for scanning. Okay, so scanning electron microscope and transmission electron microscope. Yes, anyone has any doubt? Okay. So transmission electron microscope and scanning electron microscope. So by the name itself, you can see in this case, the electron source, from the electron source, the electrons are transmitted through the sample. So this is the sample here. We have the sample here. The electrons are transmitted through the sample, and then there is a screen or a detector that detects and forms an image of the sample. Okay, so it can, since it is passing through the sample, you can see the intercellular structure. Whereas in scanning electron microscope, the electrons are deflected from the sample. So electrons hit the sample and then they get reflected back. So they are either collected by a secondary electron detector or the backscatter electron detector, and then the images form. So in scanning electron microscope, only the surface can be visualized, whereas in transmission electron microscope, the intercellular structures can be visualized. So this is a structure of a phospholipid bilayer. So you can see the cell membrane is clearly visible with the intercellular space. So the whole structure is visible. If you would have seen this on scanning electron microscope, only the surface would have been visible. Okay, only the top surface would have been visible. So transmission electron microscope uses a beam of electrons and magnets instead of lights and lenses, and an optical electron microscope to generate images of the specimen. So generally you have seen the microscopes with a light bulb that can give the light to the sample, and it's basically uh, that those microscopes are called optical microscopes. We have lenses and we have lights. In this case, instead of a lens, you have magnets which can focus the beam. So in an optical microscope, you use lenses to focus the light, right? So similarly, in this case, we have electromagnets that can focus the electrons, and we have the electron as the source, as the illuminating source instead of normal visible light. Okay, that is clear to everybody? Everybody understood this? Anybody has any doubt in this? Please ask. If you have any doubt, please write it in the chat box, okay? Okay. Moving on to the next question. Match the appearance of a gram-negative bacteria after the following steps in a gram staining. Match the appearance of a gram-negative bacteria after the following steps in a gram staining. Crystal violet, alcohol acetone and safranin on one side, and you have purple, pink to red and colorless on the other side. You have to match the left column to the right column based on the appearance of a gram-negative bacteria after the following steps in gram staining. Write your answers in the chat box. If Smita says option A. So option A means after crystal violet, the cells will be colorless. After acetone alcohol step, the cells will be purple, and safranin pink to red. Okay. What about others? Read the name of the dyes and try to at least guess. Dilip Kumar says option D. After crystal violet, it would be pink. After alcohol acetone, it will be colorless, and saffron, it will be purple. Okay. We have one more answer, option C. After crystal violet, it would be purple. After alcohol acetone, it would be colorless. After safranin, it would be pink. That seems to be the correct answer, option C. So by the name itself, you can see it has violet in its name. So it will stain the cells purple or violet in color. Alcohol acetone is a washing step. So I'll go through the gram staining procedure once so you will understand. And safranin, I told you, it is a pink red colored dye. So it stains the color the cells pink. Okay. The correct answer would be option C, crystal violet purple, alcohol acetone colorless, and safranin pink. So how do we do gram staining and what are gram negative and gram positive bacteria? We'll discuss. So basically in gram staining, what we do, we first uh, we we first stain the cells with crystal violet. We take the bacteria, like let's say we are taking a small amount of culture. We are making a smear on the slide. So you have a glass slide. You make a thin smear of that bacterial cells. Then you heat fix it. Then you heat fix it so that these cells adhere to the glass slide and they are now dead and they are fixed on the slide. Okay. Then after that, you first stain it with crystal violet. So it will stain all the cells purple. Then you wash it with alcohol or acetone. You wash it with alcohol or acetone. So if it is gram negative, it will turn colorless. If it is gram positive, it will not lose the crystal violet. Okay, so after alcohol, acetone, you will stain with safranin. Okay, so after crystal violet, actually there is one more step. So after crystal violet, there's grams iodine to fix the crystal violet, and then comes the alcohol. Okay, and then finally we stain it with safranin. So if the cells will lose crystal violet, they will get stained by safranin and appear pink. If the cells will not lose crystal violet, they will still be violet in color or purple in color, and they will then and therefore it will not be able to take up safranin, and it will not be uh, pink, it will be purple only. Okay, so this is how gram positive and gram negative bacterial cell wall looks like. So gram positive cells, so gram positive cells has a thick peptidoglycan layer. This is the major difference. Okay, and the gram negative ones have a thin peptidoglycan layer. Now, because the gram positives have a thick peptidoglycan layer, what happens when we stain with crystal violet, it penetrates this peptidoglycan layer, and when we wash it with alcohol, it doesn't leave the cells completely, and the cells remain purple in color. Okay, whereas when we wash these gram negative cells with alcohol, it loses the crystal violet easily. Since the peptidoglycan layer is very thin, it loses the crystal violet completely, and they become colorless. Okay, after alcohol wash, gram negative cells become colorless, and when after that we'll stain with safranin, so they will appear pink in color. Okay, so if we have a mixture of gram positive and gram negative, and if we observe under a microscope, you will see purple colored gram positive bacteria and pink colored gram negative bacteria. Okay, so gram negative cell walls have a thin layer of peptidoglycan in the periplasmic space within its inner and outer lipid membranes. So cell walls take up the crystal violet dye, appears purple. Then dye and mordant complex made by iodine is easily removed from the peptidoglycan layer with alcohol, making the cell wall colorless. And then this colorless cell wall easily take up the counter stain, that is safranin, and it appears pink to red. So basically we take the cells, we heat fix it, then we stain with crystal violet. Then we add grams iodine, which is a iodine solution. We add grams iodine, and this grams iodine kinds of fixes this crystal violet on the cells. Okay. Then we wash it with alcohol. When we wash it with alcohol, since gram negative has a thin peptidoglycan layer, the crystal violet comes out. Okay. But from gram positive bacteria, the crystal violet cannot come out, and after alcohol also they appear purple. Okay. And then after alcohol wash, we add safranin. We add safranin. So since these cells did not lose the crystal violet, they will keep appearing purple. But since these cells have lost the crystal violet, they will be colorless. And then if you stain anything colorless with another color, they will take up that color, right? So they will take up the pink color. And these cells are already stained, so they won't get stained by safranin. Is that clear? Gram staining is a very important topic, and it's a very important staining technique to distinguish between bacteria. If you are trying to identify a bacteria, it is a primary identification feature with what is its gram status. Okay, whether it is gram positive or a gram negative bacteria, it's a very, very basic identifying feature. So you should understand what gram staining means, and you should be able to understand the procedure of gram staining as well. Okay, so all these staining steps are done for 30 seconds to 1 minute. We add the crystal violet on the slide, we keep it for 1 minute, and then we drain it off, wash it with water. Then we add the grams iodine, keep it for 1 minute, drain it off, wash it with water. Then you add alcohol, and then like for 30 to 60 seconds, and then again remove the remaining alcohol with water, and then add saffron and keep it for 1 minute, and then again wash it with water. Okay, so this is how we do the gram staining. Moving on to the next question now. Which of the following pairs is mismatched? Bacterial identification, gram staining, dark field microscopy, light passes through the cell. Option C is three-dimensional images, atomic force microscopy. Option D is immersion oil, prevents refraction of light and its scattering away from the lens. Which of the following pair is mismatched? Bacterial identification, that is gram staining, dark field microscopy, light passes through the cell, three-dimensional images, atomic force microscopy, and immersion oil, prevents refraction of light and its scattering away from the lens. What is the correct answer? I have one answer in the chat box, option B. What about others? Yeah, another answer is option C here in the chat box. Okay. What about others? Anyone else wants to answer? Okay, we have two votes to option B. So option B is the correct answer. Dark field microscopy and light passes through the cell. That's a mismatched pair. So I told you gram staining is a primary step for bacterial identification. So that is a correct match. Atomic force microscopy, it's a technique and it is used for structure determination. So three-dimensional images can be obtained from atomic force microscopy. So that is also correct answer. Then we have immersion oil. That is also correct match. Immersion oil is used to prevent the refraction of light and it's scattering away from lens. So we put a drop of immersion oil and then we use the 100x objective lens. So the wrong pair is dark field microscopy. Light passes through the cell is not what dark field microscopy is. So dark field microscopy basically the samples uh, are the samples reflect the light away and they appear light against a darker background. So dark field microscopy, it uses a special condenser with an opaque disc that blocks the light from entering the objective lens directly. Okay, so light reflected by the specimen enters the objective lens and then specimen appears light against a black background. So it will be completely dark background and you will have transparent looking structures appearing. Okay, so those cells are reflecting the light and it is the reflected light enters the objective lens. Okay, so dark field microscopy is used for observing the cells and in a darker background. Is that clear to everyone? Any doubt? Okay, then we'll move on to the next question. Which of the following is not accomplished by fixing bacterial cells to a slide? Cell morphology are preserved, option A. Option B, cells are killed. Option C, cell structures are differentiated. Option D, cells are attached to the slide. Which of the following is not accomplished by fixing bacterial cells to a slide? Option A, cell morphology are preserved. Option B, cells are killed. Option C, cell structures are differentiated. Option D, cells are attached to the slide. Which of the following is not accomplished? Okay, pay attention to the question. What does not happen when you fix the cell? We have one answer, option B. What about others? Anyone else wants to answer? So the correct answer is option C. Cell structures are differentiated. So when we fix the bacterial cells, I told you during gram staining, we do the heat fixing of the cells. So the cells adhere to the surface. So cells are attached to the slide. That is option D is the correct answer. The cells are also killed. I told you the cells die when we do the fixation. That is how we preserve the cells as it is in a state. It's like capturing the cell, how you capture a picture. So when you capture a picture, when you take a photo from a camera, you are taking the snapshot of that particular time. Okay, so whatever is happening, so in the photo nothing is moving, nothing is uh, alive in that photo. Okay, so it's a picture. Okay, so for when we are observing things under the microscope, we also want to take picture how it is looking. But if we don't fix the cells will keep on moving on the slide. So it will be difficult for us to visualize. So we do the fixation so that the cells attach to the slide and cells are killed, but in a way that the morphology is preserved. When we do the fixation, the cell morphology is preserved. So if the cell is spherical, you will see the spherical cells. It's not like they will burst open. They will be killed, but they will be stuck to the slide as it is. The cell morphology will be preserved, only the cell structures won't be differentiated. So The internal structures won't be very clear, but the cell morphology will be uh, as it is. Okay, so cell fixation attaches the cells to the slide so that they can be used for microscopy or any other analysis. It allows most cell morphology to be preserved but does not allow the cells to be observed in living state. Okay, that's clear to everybody. Moving on to the next question then. Suppose you observe that bacteria are moving in your gram stain, you can conclude that option A, the bacteria are gram negative. Option B, the bacteria have capsules. Option C, you did a flagella staining and not a gram stain. And option D, you didn't fix the smear. Suppose you observe that bacteria are moving in your gram stain. You can conclude that option A, bacteria are gram negative. Option B, the bacteria have capsules. Option C, you did a flagella staining and not gram staining. And option D, you didn't fix the smear. What do you think would be the correct answer? Okay, we have two answers in the chat box already, option C and option D. What about others? Anyone else wants to answer? Anybody knows the answer? Okay. Option D, we have two people answering option D. Option D is the correct answer. You didn't fix the smear. So I told you fixing smear attaches the cells to the slide. Okay, so fixing smear to slide kills the cells and adheres them to the slide. Moving bacteria in slide proves lack of fixing. So if you don't fix the cells properly, you will see the cells floating around in the stain instead of uh, attached to the slide in one place. Okay, so it will be difficult for you to take a image in a microscope. This is clear. Moving on to the next question now. The ability of a lens to distinguish two points at specified distance apart is termed as. The ability of the lenses to distinguish two points at a specified distance apart is termed option A, magnification, option B, resolution, option C, numerical aperture, and option D, none of the above. The ability of the lenses to distinguish two points at a specified distance apart is termed as option A, magnification, option B, resolution, option C, numerical aperture, option D, none of the above. What would be the correct answer? Okay, we have two answers in the chat box, C and B. What about others? Ability of lenses to distinguish 2 points at a specified distance apart is termed as magnification, resolution, numerical aperture, or none of the above. We have two answers in the chat box. Option C, numerical aperture, and option B, resolution. Okay. Anyone else wants to answer? What do you think would be the right answer? Mhm. Okay, so right answer is option B. So you were correct. So option B, resolution. Resolution is a function of wavelength used in numerical aperture of the objective lens. It is ability to distinguish 2 points separated by a minimum specific distance. So basically, when we have a lens and when we are observing an image, what is the resolution basically is the distance at which you can see 2 points as separate 2 points. Okay, so if I have these 2 points separated by certain distance, okay, if I have if I have these 2 points, so if you are able to see these 2 points as distinct 2 points, that means the resolution is this much distance. If you can see these 2 points as a single point, that means the lens or the microscope does not have the resolution to resolve these 2 points separately. So the distance will be bigger than this. So the resolution will be larger. So the smaller that resolving distance will be, the better resolution you will have. The resolution of most common optical microscopes is around zero point 0.2 microns, which means that objects separated by minimum distance of zero point 0.2 microns means the distance between these 2 points, if it is zero point 0.2 micron, then also we can see the points distinctly. If the distance between these 2 points is less than zero point 0.2 micron, if it is zero point 0.1 micron, we cannot see these 2 points distinctly. We will see it as one single point. Okay. So if the separating distance is less than zero point 0.2, the object lens cannot be resolved as multiple objects and will look like a single object. So basically, these 2 points will not look like two separate points, they will look like a single point. But if these 2 points have zero point 0.2 micrometer distance, then we can see it as two different points. So you must have heard about the term resolution in terms of photographs also. We generally take pictures and we see the resolution is not good. If we are watching a movie, we sometimes say the resolution is not good. We can see blurred images. So that blurred images is basically we are not able to distinguish between two different points. So if there are two people in the scene and we cannot see those two people separate as two separate person, we can see just blurry image of uh, something. We cannot distinguish whether there are two people or three people standing there. So that means the resolution is bad, we say, right? The basically that term resolution means the distance which can like the distance between any 2 points that could be easily that could easily be visible, that could be distinguished by a uh, microscope or by a lens. Okay, so if it's if you are talking about photograph or camera lens, then that is also having the same principle like a microscopic lens. Okay, So these are the terms that we generally use in our daily life. So you should understand the meaning of it also. So everybody understood what resolution is? Anybody has any question regarding this? Okay, then we'll move on to the next questions. Match the following macromolecules in prokaryotes with their location. The locations are given as cytoplasm, nucleoid, cell wall, and cytoplasmic membrane. And we have macromolecules on the right hand side: DNA, peptidoglycan, lipids, and proteins. So we have 4 locations and 4 macromolecules. So everybody try to match the macromolecules with their specific location. Come on, fast. Match the following and then tell me the answer in the chat box. No one is able to solve this. Okay. Option B. I have one answer in the chat box. Option B. That is cytoplasm is matching with peptidoglycan, nucleoid with lipids. No, option B is not the correct answer. What about others? Anyone else? I just talked about bacterial gram staining. What was it based on? How were we able to distinguish between gram positive and gram negative? What was the major structural difference between two? Just explained you what is gram staining and how the gram staining procedure distinguishes between two. Yes, thick peptidoglycan. Where the peptidoglycan layer was present? What part of cell the peptidoglycan was present? Yes, then why did you match cell wall with proteins? You only answered option B. Did you look at the options or you just guessed the answer? And what about others? Option C. Cytoplasm with protein, nucleoid with DNA, cell wall with peptidoglycan, and cytoplasmic membrane with. What about others? No one else knows the answer? So option C is the correct answer. You have cytoplasms, you have proteins present in cytoplasm, you have DNA present in nucleoids. So I told you in the previous class, we have two types of cells, prokaryotes and eukaryotes. In prokaryotes, we don't have membrane-bound organelles. We have the DNA located at one location. That location is called nucleoid, but we don't have any membrane bound structure present around the DNA, whereas in eukaryotes we have nucleus, which is a membrane bound structure inside which DNA is present. So in prokaryotes we have nucleoid in where DNA is present. Then we have peptidoglycan, which is present in the cell wall. I told you the cell wall comprises of peptidoglycan. In gram positive, that peptidoglycan is a thick layer, and in gram negative, the peptidoglycan is a thin layer. Then you have the membrane. Membrane is composed of lipids. So we have phospholipid membrane. So membrane is composed of lipids. Okay, so the option C is correct answer. Cytoplasm with protein, nucleoid with DNA, cell wall with peptidoglycan, and cytoplasmic membrane with lipids. Clear to everybody? Anyone have any doubt in this? Try to read the question properly and read the options properly before answering. Otherwise, when you are answering these questions in an exam, you will already be very stressed out because it's an exam, and you will like it will be your tendency to give the wrong answers always. Okay, so try to be calm, try to read the question properly, try to read the options properly, and then answer. Okay, take your time while reading the question and the options, because most of the time the multiple choice questions tend to be confusing, and if you get confused and if you tick the wrong answer, it will cost you your marks. Okay, so read it properly. Moving on to the next question now. Ribose and adenine can be found in option A, ATP and RNA, option B, proteins and lipids, option C, DNA and RNA, and option D, ATP and DNA. Ribose and adenine can be found in. Okay, we have one answer, option A, ATP and RNA, and second answer is option C, DNA and RNA. Okay. Anyone else wants to answer? So the correct answer is option A, ATP and RNA. That is correct. So adenine is a nitrogenous base. Ribose is the sugar here. Okay. So ATP is a nucleotide that consists of three main structures: nitrogenous base, adenine, and the sugar. So ATP stands for adenine or adenosine triphosphate. Okay, so this is adenosine triphosphate. So we have three phosphates here. It is attached to a sugar, so it becomes adenosine triphosphate. And uh, this sugar here, it is the ribose sugar. So there are two types of sugar present, ribose and deoxyribose. So this is ribose where the 2' OH is present, and when the 2' OH becomes H, then this is deoxyribose because the oxygen is removed from the second carbon. Okay, so this is deoxyribose. So this deoxyribose is present in DNA and ribose is present in RNA. So RNA is called ribonucleic acid, and DNA is known as deoxyribonucleic acid. Okay, so since deoxyribose is present in DNA, it is called deoxyribonucleic acid, and ribose is present in RNA, so ribonucleic acid. And nucleic acid because this is a nucleotide that is present. So nitrogenous base and the sugar uh, and then phosphate. Okay. So ATP is a nucleotide that consists of three main structures: nitrogenous base, that is adenine, sugar, that is ribose, and chains of three phosphate groups bound to ribose. The RNA is ribose nucleic acid and contains ribose as well as adenine along with the other nitrogenous base such as guanine, cytosine, and uracil. Okay, so thymine is not present in RNA, instead uracil is present. Okay, that's clear to everybody. Okay, let's move on to the next question then. True or false? First statement is nucleic acids are found in storage granules. Second statement is proteins are found in cytoplasm, cytoplasmic membrane, cell walls, and flagella. For both the statements, you have to tell true or false. First statement is nucleic acids are found in storage granules. Is it true or false? Then second statement is proteins are found in cytoplasm, cytoplasmic membrane, cell walls, and flagella. What would be the correct answer? Write it in the chat box. Okay. I have one answer, option A. That is, first statement is true, second is false. Okay. What about others? Do you agree with option A or do you have any other option? Do you think other option is correct? Okay, we have another answer, option D. First statement is false, second statement is true. Okay, what about others? Anyone else wants to answer? So option D is the correct answer. First statement, nucleic acids are found in storage granules. That is false. Nucleic acid is found in, so nucleic acids are part of DNA and RNA. Okay. So DNA is found in the nucleoid region and RNA is found in cytoplasm in forms of mRNA, and it is also found in ribosomes in form of ribosomal RNA. Okay, so in case of eukaryotes, you will have ribosome. So first statement is false. Nucleic acids are not found in storage granules. So storage granules generally comprise of polysaccharides and lipids. Second statement is true. Proteins are found in cytoplasm, cytoplasmic membrane, cell wall, and flagella. Proteins, I told you, they contribute to 55% of the dry weight of a cell. So they are present almost everywhere: cytoplasm, membrane, cell wall, flagella, everywhere. Okay, so that statement is true. That's clear to everyone. So nucleic acids are found in cytoplasm of the microbes as DNA, or in the nucleus in case of eukaryotes, or RNA. Storage granules contain carbohydrates or other substrates that may be needed by an organism when the substrate or the food in the environment is scarce. So they store these nutrients, carbohydrates, and inorganic compounds as like backup food source. Okay, so these are storage granules. So that's like a storeroom for them. That's clear to everybody? Okay, we have another match the following question. Match the following monomers to their polymers. So we have monomers on the left hand side, sugar, amino acid, nucleotides, and fatty acids. And we have polymers on the right hand side, nucleic acid, polysaccharides, lipids, and proteins. Match them. Come on, answer. Okay, option B. We have one answer, option B. Sugars to polysaccharides, amino acids to lipids, nucleotides to proteins, and fourth is fatty acids to nucleic acids. Okay, what about others? Anyone else? Can anyone else answer? Option C. We have another option answer, option C. Sugars to polysaccharides. 2 is for amino acids to protein. Nucleic acids, nucleotides to nucleic acids, and then okay. And then two more people have answered option B. Option C is the correct answer. So whoever answered option C is right. Option B is not the correct answer. So sugars are the building blocks of polysaccharides. So polysaccharides, the name saccharides comes from sugar. Then amino acids are the building blocks of proteins. So amino acids are joined together with the peptide linkages to form proteins. Okay, so amino acids like glycine, alanine, leucine. So all these amino acids, they are joined together to form long chains, which are called proteins. These chains then get folded into proteins. Then we have nucleotides. I told you nucleotides are the building blocks of nucleic acids. I showed you a nucleotide that is ATP. So it had a nitrogenous base, a sugar group, and a phosphate backbone. Okay. So these nucleotides are joined together to form nucleic acids. Then we have fatty acids. So fatty acids, they are the building blocks of lipids. So lipids are made of fatty acids that are connected to glycerols. Okay. This is clear to everyone? Everybody who are answering option B, do you understand? Okay, moving on to the next question. Which of the following is not a function of nucleotide? Which of the following is not a function of nucleotide? Okay, you have to look for the option that is not a function of nucleotide. Participate in oxidation reduction reactions in cells, carry sugar in biosynthesis for biosynthesis of polysaccharides, regulate or inhibit or stimulate activities of certain enzymes, and provide structural integrity to cells. Which of the following is not a function of nucleotide? Participate in oxidation reduction reactions in the cells, carry sugar in biosynthesis of polysaccharides, regulate, inhibit, or stimulate activities of certain enzymes, and provide structural integrity of cells. We have one answer, option A. Participate in oxidation reduction. Okay, you think that's not a function of nucleotide? Anyone else? Option D. Provide structural integrity to cells. Okay, anyone else? Okay, option C. Regulate, inhibit, or stimulate the activities of certain enzymes. So the correct answer is option D. So Ganesh has answered it correctly. Provide structural integrity of cells. So you all know the structural integrity to cells are provided by the cell wall. So cell wall gives the structural integrity to cells. And in case of mature cells, you have cellulose and lignin that forms the secondary cell wall, which gives the rigidity and waterproofing. But primarily, cell wall gives the structural integrity to all the cells. But all the other functions, they are actually functions of nucleotides. So ATP and GTP, these are very common. So ATP and GTP, these are very common nucleotides. They are involved in oxidation reduction reaction. They can transfer their phosphate. They can get converted from ATP to ADP. They can get reduced from ATP to ADP and give their phosphate to another molecule. So let's say we have glucose. So glucose can become glucose 6 phosphate by the help of ATP. So oxidation reduction reactions are carried out by nucleotides. I told you nucleotides have a sugar backbone. So that sugar can be used in the polysaccharide biosynthesis. So they carry the sugar in the biosynthesis of polysaccharides, and they regulate, inhibit, or stimulate activities of certain enzymes. So we have certain enzymes that are activated or regulated by these ATP molecules or GTP molecules. Okay. So option D is the correct answer. That is not the function of nucleotide. Nucleotides have nothing to do with the structural integrity. They are part of the nucleic acids and they are part of these reactions. Okay. That's clear to everybody. If you have any doubt in any of the questions, please ask in the chat box. Moving on to the next question. Question number 17. Most abundant substance in cells is option A, protein, option B, water, option C, sugar, option D, none of the above. The most abundant substance on cells is, okay, I have one answer, option B, another option A, protein, water, sugar, none of the above. Make sure the question is most abundant substance. Okay. Most abundant substance in the cell is. Anyone else wants to answer? So the correct answer is option B. So remember that I told you the protein is present in most abundant form in the dry weight of the cell. So 55% of the dry weight of a cell was protein, but 70% of the total mass of the cell is water. So dry weight of the cell is basically 30%. So cell comprises of 70% water. Okay. So other than that, 30% has, from this 30%, 55% has proteins and then rest other macromolecules and everything. But major substance in the cell is water. Okay. Is this clear to everybody? Do you understand this? Right. Moving on to the next question. Name the following according to the shape and arrangement of bacteria. So you have two figures given here. Name the following according to the shape and arrangement of the bacteria. Option A is first is Diplococci and second is Streptobacilli. Option B is first is Staphylococci and second is Streptobacilli. Option C is first is Tetrad and second is Coccobacilli. Option D is first is Streptococci and second is Diplobacilli. Okay, we have one option. Okay, we have two answers as option B. Two people have answered option B. What about others? Look at the figure. Tell me which option is correct. So option B is the correct answer. You are right. So these are the different shapes that have been named. So if you have one spherical, so if you have one spherical shaped bacteria, it's generally called cocci. Okay. So cocci or coccus is the term that is used for the circular shape, round shape. Okay. Round shaped bacteria are called cocci. So if there are two round shaped bacteria joined together, they are called diplococci. Okay, so two, if the morphology is like this, they are present like bunch, like they are present in a pair, it's called diplococci. If they are present in fours, it's called tetrad. If they are present in eight, it's called sarcina. And if they are present in long chains of cocci, it's called streptococci. If it is present in bunch of uh, cocci, then it is called staphylococci. Okay. Same thing is with bacillus. So now if it is a rod shaped bacteria, it's called bacillus. Okay. If it is a round shaped bacteria, it's called cocci. If it is a rod shaped bacteria, it is called bacilli. So cocci is plural, coccus is singular. So bacilli is plural and bacillus is singular. So if it's a rod shaped bacteria, it is bacillus. Okay. So it is rod shaped. Okay. So two bacillus are there, so diplobacilli. Chain of bacillus is there, so streptobacilli. So term strepto is used for chain, term diplo is used for two. Then if you have cocobacillus, so you have another shape called cocobacillus. So it is round but rod. Okay. So when it is rounded and rod shaped, it is cocobacilli. Okay. If it is completely round, it is coccus. If it is completely rod shaped, it is bacillus. But if it is round and rod shaped, it is cocobacilli. Okay. This is clear to everyone? Okay, I'll move on to the next question. The plasma membrane is considered a barrier to the plasma membrane is considered a barrier to the environment because option A, it is made of structural proteins. Option B, it is a rigid structure. Option C, no molecules may pass through it. And option D, it controls the passage of molecules into the cell. Plasma membrane is considered a barrier to the environment because option A, it is made of structural proteins. Option B, it is a rigid structure. Option C, no molecules may pass through it. And option D, it controls the passage of molecules into the cells. Okay, we have two answers. Both of them are option D. What about others? Anyone else wants to answer? So option D is the correct answer. You are right. So it controls the passage of molecules into the cell. So plasma membrane, it is a semi-permeable membrane. So it is permeable to specific items but not permeable to other items. Okay, so semi-permeable membrane made up of phospholipids. These phospholipids they have amphithetic properties. The polar head groups, that are glycerols of the phospholipids, are hydrophilic and face the water, and non-polar tails, that are the fatty acid tails, are hydrophobic and they face each other, forming a practically impermeable layer through which only gases can pass. Even water cannot pass through this plasma membrane. Okay, so this is a semi-permeable phospholipid bilayer. So you can see the polar head groups made with black color and the fatty acid chains that are shown with the lines, straight lines, and you have certain proteins that are embedded in this membrane. Okay, understood? Okay, I'll move on to the next question then. Question number 20. Active transport is different from facilitated diffusion because active transport requires option A, osmotic pressure, option B, a lipid bilayer, option C, energy in the form of ATP, and option D, a transporter protein in the plasma membrane. So for this question, you have more than two options correct, okay? So more than one option correct, sorry. So there can be two options that are correct. Active transport is different from facilitated diffusion because active transport requires osmotic pressure, lipid bilayer, energy in the form of ATP or transporter protein in the plasma membrane. Okay, we have one answer is option C, another answer is option B and C. Anyone else? Anybody else wants to answer? So option C is one of the correct answers. So both of you are right there. The second correct answer is option D. So the process of moving molecules across the cell membranes using cellular energy in the form of ATP is called active transport. While facilitated diffusion or facilitated transport is a spontaneous passive transport of molecules or ions through the membrane mediated by proteins. Okay. So in case of active transport, you need energy in the form of ATP, and also you need transporter proteins. So there are transporter proteins that are dependent on ATP. So ATP will give them the energy to transport the molecules in and out of the cell membrane. Okay. Do you understand this? Hopefully this is clear to everybody. Okay. Moving on to the next question. Which of the following linkages are present in cellulose? Which of the following linkages are present in cellulose? Option A, alpha 1,4 glycosidic bonds between D-glucose molecules to form straight chains. Option B, beta 1,4 glycosidic bonds between D-glucose to form straight chains. Option C, alpha 1,6 glycosidic bond between D-glucose to form branches. And option D, both A and B. Which of the following linkages are present in cellulose? Which of the following linkages are present in cellulose? Alpha 1,4 glycosidic bonds between D-glucose molecules to form straight chain, beta 1,4 glycosidic bond between D-glucose to form straight chains, alpha 1,6 glycosidic bond between D-glucose to form branches, or both A and B. What kind of linkages are present in cellulose? Okay, we have option D as the options. Okay, two people have answered option D. What about others? Option D is both A and B. Okay. Anyone else? So the correct answer is option B. Beta 1,4 glycosidic linkages are present in cellulose. Okay. Cellulose are the straight chain molecules. So here you can see these are the types of linkages. So alpha glucose, so if I number these carbon, so this will be the first carbon, this will be the second carbon, then third carbon, fourth carbon, fifth carbon, and sixth carbon. Okay. So alpha glucose is when the first and the second carbon, both the OH are in the same side. Okay. So this is called alpha glucose. Okay. So in these both of them, you can see the OH groups are on the same side, on the first and the second carbon. So this is the first, this is second, this is third, this is fourth, this is fifth, and this is sixth. And the linkage is between the first and the fourth carbon. So that is why it is called alpha 1,4. So alpha from the alpha glucose, and 1,4 because first and fourth carbon are joined together. Okay. So first and fourth carbon are joined together. So that's why it says alpha 1,4 glycosidic linkage. When it is beta 1,4 glycosidic linkage, you see this is a beta glucose because the OH is on the opposite side. So this is the first carbon, second carbon, third carbon, fourth carbon, fifth carbon, and sixth carbon. Again, you have here first carbon. So this is beta. Okay, first carbon, second carbon, third, fourth, fifth, and sixth. Okay. So you can see again the linkage is between first and fourth. So beta 1,4. So beta from the beta glucose, and 1,4 because first and fourth carbon are joined together. So this is a beta 1,4 linkage. So this is the linkage that is present in cellulose. Okay. And then you have another type of linkage, glycosidic linkage, that is alpha 1,6. So this is alpha glucose, you can see from here. And this alpha glucose, this one first carbon is attached to the sixth carbon. So this is first, second, third, fourth, fifth and sixth. Okay, so this is alpha 1, 6. Okay, there are three types of glycosidic bonds. This is clear? Which of the following nitrogenous base is a purine? Which of the following nitrogenous base is a purine? Option A, guanine. Option B, thymine. Option C, cytosine. Option D, uracil. Which of the following nitrogenous base is a purine? I have told you this in the last session also about purines and pyrimidines. Which of the following nitrogenous base is purine? I have one option in the chat box, option D. What about others? Anyone else wants to answer? The correct answer is option A, guanine. So you have pyrimidines and purines. This is based on the ring that is present. So if it is this kind of ring, this is called a pyrimidine ring, and modifications on the pyrimidine give rise to cytosine, thymine and uracil, and modification in the purine ring, which is double ring structure, gives us adenine and guanine. So adenine and guanine are purines and cytosine, thymine and uracil are pyrimidine. Is that clear to everyone? Okay, moving on to the next question. Membrane proteins that transport substances from one side to the others are called. Membrane proteins that transport substances from one side to the other are called. Option A, antiporters. Option B, symporters. Option C, uniporters. And option D, none of these. Antiporters, symporters, uniporters, or none of these. Okay. Anyone else wants to answer? So option C is the correct answer, uniporter. So these are the three types of transporter proteins that are present. So uniporter, by the name, you can see it as a unidirectional transport protein. So it transports the substance from one side to another. Symporter transports a substance along with another carrier molecule. So they will both be transported together in one direction. And antiporter is one substance will be transported from outside to inside, and at the same time another substance will be transported from inside to outside. Okay, so that is antiporter because the directions are opposite, so anti. Okay, symporters when they are transported together, and uniporter only one substance is transported from in one direction. Okay. Okay, coming to the next question. Arrangement of flagella where tuft of flagella is located at one end of the cell is called. Option A, monotrichous. Option B, amphitrichous. Option C, lophotrichous. And option D, peritrichous. Arrangement of flagella where tuft of flagella is located at one end of the cell is called monotrichous, amphitrichous, lophotrichous, and peritrichous. Anyone knows the answer? Monotrichous, amphitrichous, lophotrichous, and peritrichous. There are four types of arrangement of flagella. One of them has a bunch of flagella on one end of the cell, tuft of flagella on one end of the cell. Anyone? Okay, it is option C, lophotrichous. So this is the four types of arrangement of flagella. You have monotrichous, where only single flagella is present at one end. Amphitrichous, where two flagella are present, one at each end. Then you have lophotrichous, where a tuft of flagella is present at one end. And then you have peritrichous, where flagella is present all over the body. Okay. So monotrichous, amphitrichous, lophotrichous, and peritrichous. Flagella is the organelle or the structure that helps the bacteria to move. Okay. So like we have the legs to move, these bacteria, these small bacteria, they have the flagella that helps them move. Okay. So this was the last question of the session. Anybody has any doubt in any of the questions? Okay. Everything was clear. If you have any doubt, please ask. Yes, someone is raising hand. Do you have a question? Rini Venue. Okay. If there are no questions, then I think we can end this session. Okay, sure. Then we can end this session. Thank you all for joining. I'll see you in the next session. Okay.